The reason it's blue in large part is because there's very, very little material in the water. There's very little plant life in that water, and that's the result of very, very low nutrient concentrations in that water. And so that becomes a really important story when we think about microorganisms. And I will tell you that despite the water being transparent, largely transparent, that it's teeming with life. There are billions of microorganisms in a single liter of water from this ecosystem. And so after working for a number of years in the subtropical North Pacific in this low nutrient, low plant biomass ecosystem, I saw a job ad at the University of Montana at Flathead Lake Biological Station, and I knew something about Flathead Lake. It is also a low nutrient, what we call oligotrophic, low plant biomass system. It's a blue lake. For those of you that have been to Flathead Lake, it's incredibly transparent for the very same reasons that the Pacific Ocean and the North Pacific is transparent. And so I thought perhaps my previous training and methodology and some of the concepts that I've learned in the ocean might be applicable to Flathead Lake, and I thought it would help me broaden my research portfolio to expand and begin to learn a little bit about freshwater ecosystems. And so I like to think anyway that a lot of what I've learned in the ocean is transferable, hopefully, to this particular ecosystem. So for those of you that don't know anything about the Flathead Lake Biological Station, first of all, I encourage you to come up. It's an hour and a half north of where you're sitting right now. It's a nice, beautiful drive. There's a picture of the looking from the lake at the station. There are resident scientists there, including myself, that are there full time. There's staff, there's students that are there all the time. We offer summer courses as well as internships. So for those of you that are interested in the sciences and want to spend your summer, for example, doing an internship, we offer paid internships to come up and work in the laboratories with scientists, or you can take some of our summer classes. And I've got a series of brochures here if you're interested of the upcoming 2018 summer session. Feel free to take one after the lecture. And I encourage you to look into the classes. The classes that are being offered in 2018 include 1, 2, and 4 week classes, and they're from all sorts of different types of things. Some of them have to do with water, like the course I teach, it's aquatic microbial ecology, but you probably can't read all of these, but there are classes in field ecology and conservation ecology. There's one in environmental sensors, which is a brand new course that'll be, I think, really cool. There's landscape ecology and alpine ecology, stream ecology, methods in ornithology, grassland ecology, lake ecology, and even a course in drones for looking at water assessment. And so there's lots of different types of course offerings that are offered at the station every summer. And so lots of opportunity for you to, if you're interested in the sciences, to gain some exposure to both terrestrial and aquatic ecology. So please, if you're interested, feel free to contact myself or Jim Elzer, who's the director of the station. I'm sure we'll be happy to provide you. His email contact is on this brochure. So, back to the topic for today. You probably saw the title and thought, really? I'm going to sit and listen for a half an hour to somebody talk about germs, microbes? And what I'm here to tell you is that not all microbes are germs, and I'm going to tell you about a lot of microorganisms that I think you should know about that are doing really important things to keep this planet habitable. And so I thought, well, I should provide you with a definition of microbes that hopefully moves beyond the concept of germs. And so I did what everybody does. I went to Google and asked Google, what's a microbe? And Google came back and told me it's a microorganism, especially a bacterium causing disease or fermentation, which isn't exactly what I wanted. So I don't think that's right, so we need to work on that. But I think a better definition is simply it's a microscopic organism. That is an organism that's too small to be seen with the naked eye. You need a microscope to visualize it. And so what I'm going to tell you is that microorganisms include a whole diversity of different functionally and genetically distinct organisms. So they include bacteria, they include organisms we call archaea, and they include single-celled eukaryotic organisms. And I'll tell you a little bit more about some of those organisms in just a second, but it's a very diverse group of organisms. So when we talk about microbes, we're not just talking about bacteria. And I'll try and use some examples to show you how some of these organisms, what they're doing. So again, just to make the point that pathogens are the things we often think about when we think of microorganisms. We think about the things that cause diseases. We think about things that give us strep throat or food poisoning, give us the common colds or the flu, etc. And those organisms we're familiar with because we interact with them, and as a result, they, I think, leave many people feeling like microorganisms are something to be avoided. And I think, like I said, what I'm here to tell you today is that there are microbes everywhere, and they're doing absolutely critical things for this planet. And so we find microorganisms in soils, in forests, in hot springs, in hydrothermal vents, under the ice, in the ice. Every habitable space on this planet is inhabited by microorganisms. And that includes the human body. And one of the things I find remarkable is that there are estimates of the number of microorganisms associated with in or on the human body exceeds the number of human cells by anywhere from 3 to 10, a factor of 3 to 10. So that means for every, on the low end of that estimate, every one of your human cells, there are 3 microbial cells in your body. Trillions of microorganisms in each of our bodies, doing really important things to help us with digestion, aiding in immunity, etc. So microbes have been around for a really long time. And that's one of the things that I think is important for us to think about when we think about the things that they're doing on this planet. They've been on the planet for some nearly 3.8 billion years, we think. And so it's hard for me to conceive of what that means. 3.8 billion is a number I can't really wrap my head around. So I put on a military style clock, so this is from starting at 0 hours, 0 minutes, we went around 1 day. So the entire 4.6 billion years of Earth's history put onto this 1 day clock. To give you a sense of how old microorganisms are, how long they've been on this planet, and how long they have been evolving the chemistry of the planet and evolving with the chemistry of the planet. So if we look at the formation of the Earth occurring some 4.6 billion years ago, so that's where our clock starts, our day starts. At about 2 o'clock in the morning, almost 3 o'clock in the morning, the formation of Earth, the land masses, the continents, and the oceans had begun to form. And by about 5:15 in the morning, we find our first evidence of bacterial fossils on this planet. So that's somewhere about we think about 3.7. There's a lot of controversy about what that number is, but call it 3.6 billion years ago. So first microorganisms had begun to evolve, and the planet was very, very different than it is now. There was no oxygen on this planet. It was hot. There was lots of reduced gases, things. So the way that those microorganisms were growing is just fundamentally different than the way we think about the way life currently grows on this planet. And I'll talk more about that in just a second. So at about 8 o'clock in the morning, we see evidence of the first photosynthesis evolving. And photosynthesis, as I'm going to tell you in just a second, and you probably know, is a process that evolves oxygen. Early photosynthesis may not have been oxygen evolving, but certainly by noon, we know that there was enough oxygen that it evolved through the process of photosynthesis, creation of oxygen through this process, that the atmosphere had become saturated with oxygen and the oceans had gained enough oxygen that the iron that's so abundant on this planet, that when there's no oxygen present, it's in a reduced form, iron stays in solution. When there's oxygen present, it rusts. And that rust precipitates and falls to the bottom of the ocean. It collects in the sediment over geologic timescales. That sediment gets uplifted, and we see it on land, and we see these banded iron formations. So we know that gives us an approximate date of how long there's been oxygen, enough oxygen for that process to be occurring on this planet. And that happened by about noon. And that led to fundamental changes in the way that life was evolving on this planet. We refer to that as the oxygen revolution, and the most probably important thing from a biological perspective that happened is it allowed organisms like us, we didn't come along then, but allowed simple eukaryotes to come along, organisms that require oxygen for their metabolism. And so the first eukaryotes evolved at about 3 o'clock, 3:15 in the afternoon in our 24-hour clock here. By about 6:30 in the evening, there's evidence of the first sexual reproduction occurring, and the first macroscopic, so visible life, didn't arise until about almost 9 o'clock in the evening in the planet's history. And there we are, modern Homo sapiens, evolving somewhere and arising somewhere around 11 o'clock at night, 59 minutes, 59 seconds into this planet's history. And I think that's remarkable given the changes to this planet that have occurred in such a short period of Earth's history as a result of that. So microbes have been here for a long time. And as a result of that long period of inhabiting this planet, they've evolved all kinds of remarkable strategies to make a living. When I say make a living, I mean they're extracting energy from the environment and they're extracting nutrition from the environment. And they have a number of different ways of doing that are really, really difficult, I think, for us to conceive of. We're used to thinking of two major metabolic processes. Those are photosynthesis and respiration. And photosynthesis, I think we're all sort of familiar with. You walk outside, you see trees, they're green. We know something about what they're doing. They're taking carbon dioxide. They're converting carbon dioxide into cells, new plant cells to grow. And they're doing so by harvesting energy from the sun and using water as a source of what we call reductant. And as a result of that, they evolve oxygen as a byproduct. The plants aren't making the oxygen because they want the oxygen. That's a byproduct of their metabolism of photosynthesis. So we're familiar with that type of metabolism. The other type of metabolism we're familiar with is what we call aerobic respiration. And that's use of that oxygen that's been evolved through photosynthesis to consume what we refer to as organic material, the material that the plants have made through their photosynthesis. Like us, when we eat a donut, my favorite example, when we eat a donut, our bodies metabolize that donut. We use oxygen, we breathe in oxygen, we convert the sugars in the donut, we combust them, just like in an engine of a car, we combust them with oxygen. And in the process of doing so, we make carbon dioxide and water, which we exhale. We gain energy in the process of doing so. And so that process, aerobic respiration, didn't arise until there was already enough oxygen on the planet through photosynthesis for that process to occur. And microorganisms have been around a long time, as I said, long before there was oxygen or even oxygen evolving photosynthesis on this planet. And so there are all these remarkable metabolisms. And I like to think that sort of the diversity of microorganisms is best captured by looking at their modes of metabolisms. And those modes of metabolisms include things like I just described. Oxygen evolution through photosynthesis, heterotrophic consumption, like us eating the donut, they can do that, but they can also do things like photosynthesize without making oxygen. They can use different types of elements. Instead of water, they'll use hydrogen sulfide and evolve sulfur, elemental sulfur instead of oxygen. They can harvest energy from chemical elements. One of my favorites is the process of ammonia oxidation. The energy from chemical elements and fix carbon dioxide, just like a plant would, independent of light. They're not using light as the energy source. They're using the energy from the chemical elements, oxidizing that, and gaining energy in the process of fixing carbon dioxide, and using it to fix carbon dioxide, just like a plant would. They have all of these remarkable metabolisms, and probably some of those that we're most familiar with are those that are involved in the process of, for example, fermentation, which is exactly one of these processes. It's an anaerobic process in this case. There's no oxygen involved in fermentation. It's the use of yeast using sugars in the absence of oxygen and making carbon dioxide and alcohol as a byproduct of that. And so we're familiar, usually, or we have some sense of these metabolisms that microorganisms possess, but sometimes it's really difficult, I think, for us to wrap our heads around the importance of these. And I'll use a couple of examples in a second that I think hopefully highlight how some of these unique metabolisms are really important to the way the planet functions. So I'm going to talk about aquatic ecosystems. And the reason that I'm interested in aquatic ecosystems, and the reason I think you all should be interested in aquatic ecosystems, is that they're massive ecosystems. They're the largest habitats on this planet. As you, I'm sure, know, the oceans are some 70% of the surface area of this planet. They're arguably 99% of the habitable space on this planet is in water, in the oceans in particular, and by volume. And so the oceans are deep and massive, and so it makes them really interesting and important systems to study. But they're also really difficult to study. And they're difficult in part because, one, it's in water. And that inherently makes it logistically hard to sample. It's easy for us to think about sampling on land, but once we move underwater, it creates technical challenges for us, including how we can capture a water sample at some depth, for example. If I go out into Flathead Lake, which is about, call it 100 meters deep, I go out and I try and, if I want to sample water at 100 meters deep, how do I do that? How do I bring water up from 100 meters without contaminating it with the water that overlies that? And so we've had to come up with, aquatic ecologists have had to come up with unique ways of trying to sample discrete parts of the water. And those include things like sampling bottles that close at each end. They go open, when we put them in the water, they're open, and we send what we call a messenger down a string that hits the bottle and closes both of the caps on the bottle, and then we can bring it back up, and the water is relatively unperturbed relative to the water around it. And so it enables us to capture a discrete sample of water. That's just one example of what many of the logistic challenges involved in working in aquatic habitats. They're also huge. These are massive habitats, as I mentioned, and so inherently we're going to undersample them. And that's probably the single biggest source of uncertainty in global carbon budgets right now. And the way that we understand the way elements flow on this planet is our uncertainty as a result of undersampling of aquatic systems. They're fluid as well. If I go out and sample in Flathead Lake today, even if I go to the same place, the water's moving. I won't be sampling the same parcel of water that I sampled yesterday. So that inherently builds in uncertainties with regard to, or at least variability with regard to time and space. And how we understand that time and space variability is really important, I think. And then, from my perspective, one of the key things, and I think one of the real challenges about these systems, but also I think one of the things that makes them most interesting for me, most of the life in aquatic systems is microscopic. When we go outside and we look on land, we see trees and we see grasses and we see shrubs and we see animals, etc. You go to a lake and you look in the water and you don't see that. There aren't trees or visible signs of vegetation, usually, in a lake. But there's plenty of microorganisms in that lake, billions and billions of microorganisms in that lake. They're just extremely small and we can't see them. And so that creates, again, challenges in the way that we sample and study them. And so the field of microbial ecology, the study of the ecology of microbes, is really, really dependent on advances in technology. If we look at major milestones in this field, they've always followed advances in technologies, whether that's through advances in microscopes and the way that we can visualize microorganisms, or advances, as I'm going to tell you in a second, in the way that we sample their nucleic acids, the DNA. We can sequence their DNA to learn something about their evolutionary history or their genetic potential. And more recently, use of satellites. It has enabled us to visualize, as I showed you in that very first slide, ocean and land systems. And in the oceans in particular, where microorganisms are the dominant form of photosynthetic life, satellites have revolutionized our view of the distribution of microbes in aquatic systems. So just to give you a sense of how abundant these organisms are, so this is a photograph that was taken from a microscope. So I filtered, you probably can't really see it, but this is just it's about the size of a salt grain. It's a 1 millimeter by 1 millimeter by 1 millimeter cube, which is volumetrically equivalent to 1 microliter of water. And if I take that one little salt grain equivalent volume of water and filter it, put it onto, in this case, a 0.2 micron filter. So 0.2 micron, another word that doesn't really mean anything to a lot of people. A micron is 1 to the 10 to the minus 6 meters, which again might not mean anything to most people. So to put that in context, the width of a human hair is 100 microns. So this filter size is 0.2 microns. So these are very, very, very small holes in the filter that enable us to harvest microorganisms from water samples. And most of the microorganisms you're seeing, we've, in this case, stained them. And now we're looking at the fluorescence of the stain that's bound to their nucleic acids. That's not really so important. Most of these microorganisms are about maybe 0.5 microns in diameter. So they're teeny. You can fit 200 of them across the width of a human hair. These are very, very small microorganisms. In that volume, in that salt crystal-sized volume of water, there's about 1,000 bacteria from Flathead Lake. If you scale that up to, that's more than a liter, but that's pretty close to a liter bottle. If you scale that up to a typical water bottle size sample, that's about 1 billion bacteria in 1 liter of water from Flathead Lake, or from the ocean, or just about any aquatic system we can think about. Their microbes are dominant forms of living biomass in those ecosystems. And there may be hundreds or even thousands of individual species of microbes in that sample. And so often when I say this or show this slide, people think, oh, I'm never going to swim in the lake again, or I'm going to keep my mouth closed. And the vast majority, 99.999% of these microorganisms are doing critically important things for the habitat of the lake or of the ocean, and have absolutely no impact on us at all. There are very, very small percentages of microbes that are detrimental to human health. And so within every drop of water, a drop of water is about a milliliter of water, we'll say, you are likely to find about 10 million viruses. So these are non-living. We don't have to get into what viruses are necessarily, but they're non-living, but they're infectious agents. Most of those viruses are infecting bacteria in water samples. 10 million of them in a single drop of water. There might be upwards of 1 million bacteria and archaea. I'll tell you what those are in just a second. There might be somewhere on the order of about 500,000 cyanobacteria in that single drop of water. And there might be several thousand small single-celled algae or eukaryotic phytoplankton. And all of these organisms are less than 20 microns in diameter. Most of them are in the neighborhood of somewhere between less than 1 micron, we'll say, in diameter. So you could fit 100 of them across the width of that human hair. And so what I like to think, anyway, it's almost a complete food web. In a single drop of water, you've got all of the elements. You've got things that are killing things together, and as well as things that can eat things all together in a single drop of water, and which I think is kind of a remarkable thing to think about. So that in that drop of water, all of that microbial activity is largely invisible to us without high-powered microscopes. And for years, we didn't know that those microorganisms were there, and we had no concept of what they were doing. And so for many, many years, when people thought about food webs in aquatic systems, they thought about the base of the food web being algae, the photosynthetic organisms that are taking sunlight and taking carbon dioxide and making new cells. That's the base of the food web. They're making their own cell biomass using the sun as an energy source. And those things are consumed by herbivores, just like on land, deer consuming the grass, which are then consumed by some carnivore. The herbivore gets consumed by some carnivore, which ultimately then goes into fish biomass. And that, for years and years and years, was the way we thought aquatic systems worked. And I'm here to tell you that that is not the way aquatic systems work. That is one model of how aquatic systems work. But probably upwards of 60% of the carbon or the biomass in that fish, we think, probably derived from an alternate pathway that involves microorganisms. And that pathway is through this pool of stuff we call dissolved organic matter. And I like to think of it as you take a glass of water and you put a sugar cube and you dissolve the sugar cube. The sugar is still in the water, it's now dissolved. It's gone from a particle, the sugar cube, into a dissolved phase in the water. The exact same thing is happening in lakes and in the oceans, etc. And that pool of material is being introduced by photosynthetic organisms through the process of grazing things. Aquatic systems are messy places. A lot of these organisms are not very efficient eaters. They're eating cells and spilling the guts of the things they're eating, and the guts of the things they're eating, the proteins and the nucleic acids and the lipids, all that stuff enters what we would call this dissolved organic matter pool. And we think that's the stuff that in large part is fueling the nutrition and energetic demands of bacteria, of those billion bacteria in that liter of water. So those bacteria are consuming this pool of material, and in the process, it's now entered into the cellular form, which enables things to eat it again. So it's a whole other side of the food web that we hadn't previously thought about, where you go from this non-living pool of material back into a living pool of material, and then it's available for transfer back into the food web. We refer to that as what we call a microbial loop. And it turns out to be a really, really efficient, or inefficient way of moving carbon up to fish, but it turns out to be a super important and active way of moving carbon or material into fish biomass. So it's been really, over the past probably, 15 years, probably the single biggest advances in the field of microbiology and certainly in aquatic microbial ecology has come as a result of advances in DNA sequencing technologies and computational power that enable us to analyze those sequencing technologies. When we look at microscopes, or when we look at microorganisms under a microscope, it's almost impossible to tell, based on their appearances, who they are. They all sort of look like little round dots. So it's very difficult to tell from their morphology who they are, and it's almost impossible to tell what they're doing. And so, as a result of advances in DNA or nucleic acid sequencing technologies, we've begun to unravel a little bit about the role of these microorganisms in ecosystems by learning something about their genetic potential, but also about their evolutionary history. And one of the things that we've learned, and this is some famous work that was done by a scientist named Carl Woese, later followed up by a guy named Norm Pace. And what they began doing was they began sequencing specific genes that are common to all living organisms. Ribosomal RNA genes, doesn't really matter what genes they are. It does matter, actually. For this purpose, it doesn't really matter. And what they found was that, at least when I went to school, I was taught that there are 5 kingdoms in life. I don't even remember what they are now, but plants and animals. And in any case, what they found is that that's all wrong. That 5-kingdom view that at least in my generation I was trained in biology to learn is fundamentally wrong. And that in fact there are now what we call 3 domains in life. And those 3 domains are bacteria, archaea, and eukaryotes. And they are phylogenetically, that is, what I mean by that is they're genetically completely distinct from one another. And although the bacteria and the archaea are both what we call prokaryotes, they lack internal organelles, they look alike under a microscope, they're microscopic. Although they look alike, they're evolutionarily speaking completely different from one another. And the archaea in particular, originally we had thought archaea were isolated to really extreme environments. If you went to Yellowstone and sampled in the hot springs, archaea would be the dominant things you'd be sampling. If you went to hydrothermal vents, where you'd usually find them in hot places, salty places, places rich in methane, really extreme environments. And we now know that those organisms, in fact, are all over the world, including comprising some of the dominant pool of biomass in the deep ocean, which is not really an extreme environment. It's cold. It tends to be under high pressure, and but tends to have very low concentrations of nutrients. And those microorganisms tend to dominate in those systems. So The other thing that Woese and Norm Pace immediately realized when they began sequencing these ribosomal RNA genes is that the vast majority of life on this planet is microbial. There are, aside from these little on this phylogenetic tree, these little branches right here represent those things which are macroscopic, or things that we typically think of when we think of life, and the rest of this tree of life is all microscopic. And the vast majority of these organisms, they quickly realized, have never been cultivated. We have no cultivated representatives. And that might not mean much, but if you think about, if you want to study the behavior or learn something about a cat, it's hard to do that from just looking at how it aligns itself based on its nucleic acid sequence. It's nice to have the cat study it, right? For these microorganisms, we don't have, 99% of them, we have no representative to study in the laboratory. So we don't know anything about its physiology. We don't know anything about what they're doing in these ecosystems. And so that's presented an enormous challenge for microbiologists. And so through sequencing of DNA, we begin to learn something not only about who they are, but what they're genetically capable of doing, their potential, so to speak. And that's because we know by learning something about the genes, which encode proteins, and the proteins really are the machinery that make things happen. They're the catalysts that drive a lot of chemical reactions, biological reactions on this planet. And so we can begin to learn something about the continuum all the way from genes up to ecosystems. And so just to give you a little context, the human genome, our genome, all of our DNA, it's about 3 billion base pairs, A's and T's, C's and G's, 3 billion of those. There's about 20,000 protein-coding genes in our genome. And back in 1999, when it was first sequenced, it cost $300 million to sequence the human genome. You can get, I just looked a few days ago, you can get, the human genome can be sequenced now for $1,000. You could have your genome sequenced if you had $1,000, which is amazing, I think. But in any case, bacterial genomes are about 3 million base pairs, so much, much smaller genomes. So it made them much more efficient to sequence and much cheaper to sequence. And so we have a lot more information on bacterial genomes than we do on a lot of the eukaryotic organisms. And they encode about 3,000 protein, and they have about 3,000 protein coding genes. And you can get a bacterial genome sequence now for under, that's about $200. You can do the entire bacterial genome. So Craig Venter, who was one of the people that first sequenced the human genome, after he finished sequencing the human genome, he had this huge computational capacity, this huge sequencing facility that he had constructed for the sequencing of the human genome. He loved to sail, and he decided he was going to go and sample the ocean and sequence, as he referred to it, the genome of the ocean. And so he went out to a place initially off Bermuda and then conducted a whole transect, which you can see here across the world's oceans, sampling just the surface waters of the world's oceans. And in the process of doing that, and he did what we call shotgun sequencing. He sequenced every gene he could from the samples he collected in an effort to learn about the microorganisms in those samples, what they were doing, etc. He found thousands of new species that had never been seen before, more than 1 million new protein-coding genes, which was 10 times the total number that had been discovered to date, hundreds of these photoreceptors, which previously had not been known, which are a totally new way of harvesting energy from sunlight that we didn't know about. And that was from just sampling in the surface seawater only. And so people have been following up on this. This was published in Science in 2004, and so it's been more than a decade since then. People have been following up on this work, doing full ocean depth characterization, and the findings have been absolutely remarkable. So I wanted to provide just a couple of quick stories about work that's related to my own research on microorganisms. And I think one of the things that we've learned about microbes in aquatic systems is that they are often the first responders to changes, which makes them really interesting and valuable things to study, I think, because they're almost always the first things to respond. For example, in this case, when nutrients come into a lake, It's the microorganisms that are going to respond, and we can see them. This lake is green, flatted lake is blue. It's blue because of the lack of nutrients. When nutrients are introduced, the microorganisms can grow quickly, increase in their biomass, and we see them in the lake. So one of the things that I think is kind of an interesting story that relates in this case to nitrogen cycling, which is one of my interests, is that microorganisms also play a role in removing nutrients from aquatic systems. And so a classic example of this, so what's shown here is a map that shows the Mississippi River watershed. And you can see much of the watershed drains from agricultural areas as well as several large cities. And as a result of that, a lot of nutrients are introduced to the Mississippi River. So by the time the Mississippi River emerges in the Gulf of Mexico, it comes out of the Gulf of Mexico, which is a warm body of water to begin with, so it's fairly stratified. Fresh water is less dense than salt water, so the water stays on the top of the when it hits the ocean, it stays on the top of the salt water. It's full of nutrients, there's lots of light. You get a big bloom of algae as a result of that plume of nutrient-rich water coming out of the Mississippi. Well, the first thing that happens after that big bloom of algae, those cells die, they begin to sink, and as they sink, the bacteria in the underlying water begin to respire. They're using the algal cells as a source of energy, making carbon dioxide and consuming oxygen. And as a result of that consumption of oxygen, this is what they refer to as dead zones. Maybe you've heard this. They consume all of the oxygen in the underlying water, which makes it now toxic for eukaryotic organisms, fish, shellfish, etc., to live in those waters. But one of the things that the bacteria then immediately begin doing, there's a whole group of bacteria then, which thrive under those conditions. They have a unique metabolism where instead of using oxygen to consume their donuts, like they're eating a donut, no, in this case it's the sinking organic material from above, instead of using oxygen, they can use nitrate. Nitrate is one of the fertilizers that's being introduced that causes the bloom to begin with. They use nitrate, and in the process of doing so, they're respiring dinitrogen gas, they're making N2 gas, or nitrous oxide, which is another gas. They're effectively removing the nitrogen from that ecosystem. And so, in many ways, they're sort of remediators, if you will, of nutrients coming into the Gulf of Mexico. Having said that, you might have noticed I said nitrous oxide, which is also laughing gas. It's kind of a fun gas. Nitrous oxide concentrations have been going up. It's a really, really important, globally important greenhouse gas. Much more powerful than carbon dioxide as a greenhouse gas. And so the process of that, what we call denitrification, the removal of that nitrate and production of nitrous oxide, has potential climate implications. So we haven't really begun to consider that to date, but simply I guess the point is there are connections between our food needs, watering of crops through this Mississippi watershed, introduction of nutrients to those crops, those nutrients get washed out of the watershed, stimulate microbial growth in the Gulf of Mexico, which ultimately may have a climate-based connection that I think might be important to think about. One of the other things that I've been working on is I kind of like thinking about climate or teleconnections, as I refer to them. In this case, I study a lot on microorganisms that have the capability of fixing nitrogen. They can use dinitrogen gas as a source of nutrient. Most organisms cannot do that. So there's lots of nitrogen in the atmosphere. 70% of the gas in the atmosphere is dinitrogen gas. Most organisms can't use it. It's inert to us. There is a select group of microorganisms which are capable of using that dinitrogen gas in the atmosphere and converting it to ammonium, which is now a source of nutrients for them to grow on or other organisms to utilize. And it turns out these organisms have a high requirement. The enzyme that catalyzes that process requires iron. And in large regions of the world's oceans, there's very, very low supply of iron. Most iron comes into the ocean in the form of dust coming off of continents. It's blown into the ocean. for example, in this case, off the Sahara Desert, you can kind of not really see this plume of dust emerging over the ocean, settles onto the ocean, and provides a source of iron to the ocean. These microorganisms, in particular this organism here called Trichodesmium, can take advantage of that iron being introduced and begin to fix nitrogen and introduce a source of nutrients into these habitats. So I'll just tell you that I think in the next few years, the sort of future of aquatic microbial ecology, I think we're beginning to establish the parts list or the blueprint on how these systems work through sequencing of microbial genes to learn more about the function of microbes in these systems. With that, we can begin to prepare what I think of as the assembly manual, that is, understanding something about how genes are regulated and ultimately the physiology and metabolism of organisms in these systems. And then I think the real challenge over the next probably decade will be how we integrate that information beyond just the field of aquatic microbial ecology and how we begin to integrate that information into our understanding of biology, chemistry, and really what I call systems ecology. And so with that, I would encourage you to think about microorganisms as beneficial. They play a whole host of, I think, really important and unique roles on this planet. They produce and consume oxygen and organic material. They produce and consume greenhouse gases, including carbon dioxide, methane, and nitrous oxide. They're involved in nutrient recycling. They're involved in, as I mentioned before, nitrogen availability. And in essence, as Louis Pasteur said, the very great is accomplished by the very small. So with that, I'm happy to take any questions. And thank you for your attention.